The tectonic plates and fault lines over which much of the Guizhou region sits sees many earthquakes throughout the year. Studying the strengths and weaknesses of certain types of rocks helps architects better understand where to build structures to best meet the safety requirements to fit the regions. Jinfeng Zhang and company analyzed the effect of stress applied parallel and perpendicular to the structure of slate samples, as well as the role of quartz to clay ratio within each sample, which had cylindrical dimensions of 50 by 100 millimeters. The ratio of shear cracks to tension cracks was analyzed to measure the effect of stress on the rock samples and determine how many of the cracks were seen from the application of pressure in megapascals and not from the stress of removing the rock from nature, which were tension cracks. The authors concluded that the larger amount of quartz in the rock samples resulted in more brittle samples. From the testing of parallel to perpendicular strain, parallel strain to the structure increased the strength of the sample, while perpendicular strain caused reduced strength due to frictional crack expansion. Other studies have been done studying the structures of other rock types, including sandstone, red sandstone, and fractured sandstone, but no other testing has been done on argillaceous slate, which is prominent in the region.
Current measurements of the sweetness of molecules depends mainly on sensory evaluation, how humans describe their experience of the intensity of the sweetness of various molecules. Previous work missed the subtlety of how sweetness is initiated and how it varies with time. Here, Junling Deng and co-workers developed a process where they could measure the rate at which sweet receptors in living cells bind sweet molecules on the millisecond timescale. As seen in this figure, sweeteners and cells with sweet receptors are mixed in a stop-flow apparatus. Each binding of a sweetener to its receptor releases a calcium ion that leads to a fluorescent signal. The researchers found that they could divide their sweeteners into two groups. Group 1, such as xylose and erythritol, where there is an immediate rapid binding followed by a pause before binding resumes at its full rate. And Group 2 sugars, including sucrose and fructose, that have a different, more direct kinetic profile. Using their data, the researchers were able to measure the kinetics of binding and discovered a cooperative dosage dependence. This means that having more sugar amplifies the rate at which sugar molecules bind. These studies help explain the rapid onset of sweetness as the concentration of sugar molecules passes the threshold of sweet perception.
Arthritis is a chronic autoimmune disorder that leads to the breakdown of cartilage and bone due to inflammation. Many factors like age, gender, tobacco smoking, obesity, and environment have the potential to make changes in normal human protein structures, leading to the activation of the adaptive immune system, resulting in the secretion of pro-inflammatory cytokines, ultimately leading to the deformation of joints. Previously, to treat inflammatory arthritis, steroidal and non-steroidal anti-inflammatory drugs, also known as NSAIDs, have been used. The chronic use of these medications is associated with abdominal pain, ulcers, gastrointestinal bleeding, and many other symptoms. Malik and his fellow researchers have evaluated geraniol, also known as GER, a plant-derived acyclic isoprenoid monoterpene, for the prevention and treatment of inflammatory arthritis. Inflammatory arthritis was induced in rats by a single dose of complete Freund's adjuvant. After 1 week of immunization, animals were treated with methotrexate or either 25, 50, or 100 mg per kilogram geraniol. PA volume, histopathological, hematological, radiographic, and qPCR analyses were performed to analyze the severity of the disease. GER treatments at 50 mg per kilogram showed comparable efficacy to methotrexate treatment, showing a prominent reduction in PA volume and tissue necrosis. Moreover, X-ray analysis of GER-treated groups showed a reduction in the inflammation of the joints, along with no changes in bone or cartilage. It can be concluded that GER protects against CFA-induced inflammatory arthritis in rats via the suppression of pro-inflammatory cytokines, prostaglandins, and MMP1.
Decreasing levels of magnesium can be correlated to acetaminophen-induced liver diseases. However, scientists have not yet been able to create a sensor that is capable of magnesium selection over calcium binding, therefore limiting the available study of many biological pathways and body systems. Michael Brady et al. Have overcome these obstacles and worked to synthesize a new fluorescent sensor, MagZet1, that exhibits all the desired indicator qualities without the interference of calcium ions. This particular ratiometric sensor was synthesized from previously produced MagDMA, a dimethylamino functionalized sensor which failed because of its low emission quantum yield, limiting its imaging applications. MagZet1 is a quinoline-based fluorescent sensor featuring an azetidine moiety at the 6th position of the quinoline. This sensor demonstrated excellent detection of magnesium as well as selection over calcium ions. It displays a red shift in fluorescence emission, making it one of the few current sensors available for magnesium study. Other benefits include its low propensity to form ternary complexes, its dependency on free magnesium concentrations, and it does not suffer from significant pH interference. Much of this study relied heavily on flow cytometry and fluorescence microscopy to produce such results. Due to the advancements in this field, the study of many bodily systems can be further observed and re-examined in the cases where calcium detection has clouded the detection of magnesium movement and involvement. A much stronger sense of knowledge and understanding of these systems can be derived, as well as a better connection of interplay between the two cations.
The health status of Vlad Dracula the Impaler was thoroughly analyzed through a series of letters that he wrote in 1457 and 1475, allowing detection of a variety of personal afflictions and environmental conditions. Previous techniques were known to cause some damage or contamination to historical artifacts during analysis. Consulo and Associates analyzed 3 letters written by Vlad the Impaler using a new and non-invasive technique known as EVA, which stands for ethylene vinyl acetate. Their goal was to extract and study ancient proteins from these documents in order to learn more about different environmental conditions that were present during 1457 and 1475. The scientists applied strips of ethylene vinyl acetate to various parts of the letters, uplifting numerous ancient proteins and peptides. Data collection from mass spectrometry were compared to various online protein databases in order to determine which peptides were from the original time period of Vlad the Impaler and which peptides were contaminants. As a result, Consolo and his co-workers concluded that Vlad the Impaler suffered from a pathological condition called hemolacria, where he could shed tears mixed with blood. He also may have suffered from inflammatory processes of the respiratory tract and the skin. The use of non-invasive techniques such as EVA to analyze historical documents is important so that we may continue to learn more about the past while still being able to preserve ancient artifacts.